Welcome, everybody. Thank you for opening this video. My name is Tania Hernandez, and I am a research scientist at the Desert Botanical Garden. And if you're opening this video, it is likely that you sign up for a volunteer shift to help us cleaning seeds and drying seeds for the Saguaro Seed Bank. So this is a training video, a relatively brief training video. I'm going to try to explain here how we do to clean the seeds and dry the seeds. But before that, very briefly, I'm going to explain a little bit of the project and why it is important and why the task you are going to help us with is really key. So I'm going to share my screen and I'm going to share a presentation with you. So the Saguaro Seed Bank is a project part of the Saguaro Initiatives. And this emerged because of general concerns of the community about the saguaro health in the Phoenix Valley. So we realized this plant was absolutely important for the people, not only for the ecosystem. It's not only a key species for the ecosystem, but it's also very important culturally for the community. And it is a key element, icon of our region. So we decided to start several initiatives to study, better understand the saguaros in the valley and in the wild, and to help protect this icon. Some of these initiatives, you can see them listed here. Some of them you're more familiar with, for example, Saguaro Census. We have a project named Saguaros Under Stress, Saguaro Seed Bank. With the Saguaro Seed Bank, we are going to do the Saguaro Nursery and the adoption program. And we have a genetics and conservation program too. And all these projects are related. If you want to learn more, please visit our webpage, the Saguaro Initiatives website under the dbg.org site. Okay, Saguaro Seed Bank. Well, in urban spaces around the Phoenix Valley, we all notice that saguaros seem to be struggling to survive. It is very dramatic to see them dying, particularly after these strong heat waves over the summer. However, this also seems to be the case in other areas along the distribution. So to the right, you can see the natural distribution of the saguaro. It covers all the Sonoran Desert in Phoenix and in Arizona and in Sonora, Mexico. Half of the distribution, as you can see, is in Sonora, and half of the distribution is in the dry regions of Arizona. We decided to visit many sites along the distribution of the species. We sampled them first with the intention of obtaining DNA samples to analyze the DNA and better understand the natural history and the population genetics of the species. We brought these samples. What you see here is the team that went to sample to Mexico. We took little pieces of stem. We need to travel with this, which is a nitrogen tank, and we need to freeze the samples immediately to bring them to the lab. If we don't freeze the samples immediately, the DNA starts degrading, and we are not able to use the next generation sequencing methods that we are applying. So these are delicate processes, and it's really hard work, believe me. Well, we took them to the lab, we extracted the RNA and the DNA, and we performed the analysis of the sequences. And I'm not going to go into detail, but this is what we got. So this graph that you see here is a circular graph where each of the tips is a sample, a plant, and they are connected with lines. And this is analogy. The analogy of this is a genealogy, a genealogical tree. So two plants are connected because they are genetically closer to each other. So as you can see here, in green, we have the Mexican samples, and in reddish or pinkish, we see the samples from the United States. And in blue, interesting, in blue, we took samples, random samples of many saguaros we have in the garden and urban saguaros. And as you can see here, we have a very good or relatively decent representation of the genetic diversity of saguaros of the US. However, we don't have any representative of the genetic lines in Mexico. There are some genetic lines in the US that we don't have represented in the garden. So with these results, we decided to start a Saguaro Seed Bank and go travel all over the natural distribution of the species, sample fruits, and assemble a seed bank that would help preserve all these genetic lines, but also propagate all these genetic lines. So this is the sites we visited. This is an example of the sites we visited. You are going to notice that we missed some important spots, particularly in Mexico. Some of the places were not safe to visit, and we decided to be cautious and not to visit them. But we performed the best sampling that could be done with given our time frames and our resources. Those are all the spots we visited, and for each of them, we gathered DNA samples. We are visiting them again to gather fruits. Gathering fruits resulted harder than gathering DNA, than gathering samples for DNA, because when you gather a sample for DNA, you go there, you visit the plant. It doesn't matter if the plant is flowering, is not flowering, the fruits are ready or they are not. You just take a piece of the stem, freeze it, and bring it to the lab. In the case of fruits for the seed bank, it has been really complicated. Different plants along the range of the saguaro flower and fruit at different times. We tried to figure out with colleagues and friends when were the plants ready, so we were able to visit them. When we were there, some of the plants were not ready. Some of the plants, in some of the cases, the fruits were green. We harvested them anyways, and we are trying. This is the first time we do this. We are trying to figure out the best way of doing it, and we are learning. You are going to help us learn about this process. So this is an example of how we sample. We get this large pole here, and we just pull the fruits down. We catch them or we grab them from the soil or from the ground. We put them in paper bags, and then we bring them to the lab. This process can also be done at home. And this is my beautiful daughter, who is a volunteer too. And we clean the seeds. I'm going to go into more detail. What are we going to do with these seeds? Well, as I said, we are going to have a conservation seed bank. These genetic lines are going to be preserved for many, many time in the seed bank at adequate temperatures. We're going to do research with some of the seeds. We're going to try to see the best methods to keep the seeds longer. We are going to do several germination trails, but for other important part of the seeds, we're going to propagate them. We're going to germinate them. We're going to grow baby saguaros, and eventually, when they are of a decent size, strong enough, we are going to give them under adoption. And people is going to be able to adopt a saguaro from the desert, propagated by the Desert Botanical Garden, but the genetic line is already studied, analyzed, and the genetic line and the geographic origin and all the information of the plant is going to be available. So the people adopting these plants is going to be responsible to keep them alive, and this is going to be a community effort towards the preservation of a species. Okay, let's start talking about how we clean the seeds. Well, first, how we sample them, how we collect them. Well, as I said, you grab this large pole that we show here, and we put the plants in paper bags. So each plant has a number, and we have a code. It can be a 3-letter, 4-letter code of the name of the locality. For example, this is, well, you cannot see very well, but this is the plant number 06969, and this is KIN, kin, because it's Quino Bay in Mexico. This is another example. This is Saguaro Seed Bank, plant number 082, and the locality is IMU, Imuris, in Mexico. The most important thing to keep track is the number of the plant, because all the other data we have it in our notebooks or in an Excel file. But the number, you really need to keep track of that number. Don't lose that number. Well, this is how one of the methods we use to take the fruit. It is a really hard work to do. It is really hot out there. The fruits are not, the plants are really, really tall. So each fruit is really precious to us because it took us so long to dry there to find it in the right state, ripe enough, and bring it. And so we love all our plants and all our fruits. Please be careful with them. Okay, I'm gonna go through the 10 steps to clean saguaro seeds. It is very easy. Here is 2 beautiful volunteers that are helping us in Mexico. The step number 1, grab a piece of paper and grab your bag of fruits and write down the number of your plant. This is the very first plant we cleaned, and this is probably the most important step. You don't want to lose track of this number. Once that you put the number in the piece of paper, well, some of the bags, for some people that collected plants, they wrote some data in the bag, directly in the bag. And almost 99% sure that we have that information in our notes. But just in case we don't, please write down that information in a notebook, in a piece of paper, and later you bring it back to us. Okay, the step number 2 is open your bag, and each bag is going to be different because in some places we got too early, in some places we got right on time, and in some places we got late. So, for example, in these 2 examples I show you here, the fruits on the left, they are really good, perfectly ripe, ready to be opened. The fruits on the left, on the right, sorry, they are mostly old. However, they are still good, and the seeds in there within the fruits are still very good. In some cases, you're gonna find green fruits. We're gonna go over that. Open your bag, and each bag is gonna look different. Please be aware that since we were doing field work, in some cases there's some it might be bugs, ants on the bag, little pieces of I don't know, rocks or plants. So each bag is going to be full of surprises. Step number 3, select the fruits you're going to open, the ripe fruits to start. And this is just a scale of color that we put here to illustrate how is the process for the fruits. So in some cases we have green fruits. If the fruits are green, they might still have good seeds inside. So what we have observed is that the fruits start to grow, and when they reach certain size, when they are large and chubby, they start changing color, first to a yellowish color, and then to a reddish kind of color. When they are yellow, or they have yellow spots or reddish spots, they are in a perfect state. However, if they are large and chubby but green, it might be that the seeds are really good inside. In some cases, the fruits are really past and they look black, they look dark, but you open them and inside they are really good. The only case where we don't recommend to keep cleaning the seeds and keep processing them is when the fruits are black and there's mold. those fruits, unfortunately, you're gonna have to throw them to the garbage because we don't want to propagate a contamination. Step number 4 is you open each of your fruits. Well, let me tell you something that I didn't tell. If the fruits are not ready, if the fruits look green, you might want to keep them in a paper bag and let them ripe for 1 or 2 days. So just put them back in the bag, wrap them, and it's better if they are outside in the heat. Perhaps not in the direct sun, but the warmth is gonna help with the process. Now, the step 4 is you open your fruit with a knife. We generally use a cutting board, and you scoop the pulp out with a spoon and you put it in the strainer. So once you put it in the strainer, and in some cases, as you can see, I put here pieces of some pieces of fruit where the seeds are still there, but I couldn't scoop them out. So I rinse them together with the other pulp. So you take it to the water and you rinse it, and with your fingers, you try to get to push the pulp and to smash it to make it more liquid if you want. And you are trying, with the water flowing, you're trying to get rid of the most pulp you can. Step number 6, well, once that you cannot get rid of more pulp, and it is going to look like this because it's not easy to get rid of all the pulp at once, you, in your piece of paper that you previously put the number, you just flip your strainer and let all the seeds fall in the piece of paper. And then with your fingers or with a spoon, you spread the seeds to let them dry. So this, well, step number 7 is let them dry. Well, we have a video here, but let me show. Let me voltear aquí para que toda la semilla caiga aquí, okay? Okay, this is it. I didn't know this video was gonna work. I'm explaining my daughter. You see, it's really easy. It's not a big deal. Then step number 7 is you let them dry, as I said, for 24 hours. It might be less. You're gonna repeat the process. You're gonna repeat rinsing the seeds. But this step is important because when the pulp gets dry, it gets like flaky. And it's very easy to remove it once it's dry. So you let them dry. In this case, we are letting them dry inside the house because we had an ant problem. This is the Airbnb that we rented in Sonora. So we had ants and birds outside, so we let them dry inside. Later, we found a spot on a balcony where we put them in a balcony, and it was faster because it was outside. In my house, I put them in a garage, and since it is really hot, they dried faster. So you let them dry. When they dry, it's gonna look like this. And then you're gonna try, when you start moving this mass of dry seeds, you're gonna see, as I'm showing here to the right, that the pulp separates from the seeds. So you put this to rinse again. Well, very soon you're gonna find yourself with a lot of seeds and a lot of papers, and you're gonna run out of space. So plan accordingly because it might be a disaster in your house. You're gonna end up having a lot of seeds around. Well, for the ones, some people prefer to do this in your home, and that's perfectly okay for us. Or if you prefer to do it in the garden, that's also good. Okay, step number 8, if needed, please give the fruits a second rinse. Sometimes they need a third rinse. But as you can see in this image, after the second rinse, the seeds look really, really good, right? They are very clean. They still have a little bit of pulp, but that's okay. And once that they are after the second rinse, you consider that they are decently clean, you can start putting them in coin envelopes. Please be sure that your envelope includes the right number and any other data that you want to put in there. You can write down, please, the date when you clean the seeds and when you are packing the seeds in the envelope. Please include the code of the locality, but as long as you have the number, the right number, it's gonna be okay. Step number 10 is optional, but I prefer to do this. So I tape the envelope because, well, if you remember, you have seen this type of envelopes. They have some glue to tape the lid to the envelope, but the glue doesn't go all the way to the top. So sometimes the seeds are so tiny that the seeds might get out of the envelope through these little holes. So for that reason, I put this tape. And that's it. You finished. This is my other volunteer, or voluntold, my son. He's showing the last envelope he did. And we generally put them in Ziploc bags, and we keep them in the fridge. I'm going to show you, we're about to finish. I'm going to show you how many seeds we did in our second trip to Sonora. This is the second trip. Look how many seeds we packaged. The first trip we did a similar amount, so this is a lot of work. Oh, you're going to see that for some plants, you need 2 or more envelopes, and that's okay. For some plants, you need 1. For some plants, you need 2 or more. We are going to compare per locality how many seeds, what's the productivity in each of these locality. And we are going to try to correlate with the genetics. We're going to try to correlate with the temperatures and the size of the plants and other conditions. This is my other volunteer. This is my husband, and we are showing the very last envelope we did in Sonora. It is very nice to see the work all done. We were very happy doing this trip, and I really want to thank you so much for your help. You're willing to help us. When you finish, please contact me, and we can arrange to collect these envelopes with the seeds. Well, thank you very much. Thank you very much for your help. Please visit the Saguaro Initiatives webpage. And if you are working as a volunteer, I'm going to provide my personal number and my email. Please contact me, text me, send me pictures. If you have any question, please let me know. There's one thing that I forgot, and I'm going to go back here in the presentation to the green seeds. What do you do with the green seeds, with the green fruits? Well, the green fruits, you keep them, you wrap them, and you wait for them to get ripe. Sometimes they don't. They are not going to get red. They're going to get black. But that's okay. You can open them, and the seeds might be ready. If you open the fruit, it's inevitable. You need to process it. It's like avocado. If you open an avocado, it's not going to ripe. So you need to use it. Clean them anyways. But when you clean a green batch of fruits, please write in the envelope that this is a green batch. Why? It is important because we're going to compare germination rates of green batches with ripe batches. And we're going to see how many green batches in the different localities. So it is going to be an important question for us. All right. Thank you very much. This is a long video, but I hope you see it and I hope you enjoy it. And let me know if you have any questions. Thank you again, and bye-bye. Okay, bye.